In this video, we're going to go through the parameters we need to set in order to analyze our images and extract data. With our data loaded into Athena and analysis setup complete, we're going to click on the Next button. Now we are on the Analysis Parameter Calibration screen. We see a preview image here, which is the first image in our dataset. To its left, we see a number of thumbnail images, one for each color. In my particular dataset, they are the blue, corresponding to the nucleus structure, green, corresponding to the cell structure, and red, corresponding to the protein integration signal. The bottom thumbnail is an overlay image combining all of the fluorescence channels together. Below that is a definition menu where we can describe how we want our overlay to be represented. Further to the left, we see the menus containing the image analysis parameters we need to set. But before going there, I want to explore the image preview screen first. Below the image, we can see a window containing the pixel intensity histogram. This depicts the distribution of pixel intensities contained within our image. By clicking on the logarithmic scale button here, we can get a better visualization of the bright pixels corresponding to the signal of interest within the image. Through this plot, we can set the contrast for the image by dragging our mouse or cursor inside the plot until we're satisfied and get a nice looking high contrast image. As well, if we move our mouse throughout the image, we can get a readout for the current pixel intensity by looking at the blue line drawn within the histogram plot. You can see that blue vertical line change the location within the plot as I move my mouse to different locations. To the right of the histogram, we have our plate navigation menu. There, we can select different wells to be visualized. Because some wells can appear small depending on the plate layout, we can expand it using this upper right button to get a full screen view. Here we simply click on a well and can visualize the images contained within that well. Below the image preview screen, we have two arrows, left and right, corresponding to changing the field of view within the image. If we click on the drop-down menu, we can see the different options for moving within our dataset. If we have a time-lapse or Z-stack, we can move to different time or Z slices. Choosing the last option, we can also move between different wells. Below the parameter calibration box, we can see a quick access listing of all of the wells that we have previously visualized. Currently, I'm in G5. If I use the arrows, I can navigate to the neighboring well. To facilitate visualization of the preview image, I'm going to minimize the pixel intensity distribution and the plate navigation menu. Now that we're comfortable with the interface, we'll walk through the different parameters that need to be set in order to analyze our images. First, we click on the top thumbnail, corresponding to the nucleus channel. We can see that some nuclei are already identified, as indicated by these blue rings around each nucleus. These rings are the masks, and the circumscribed green rings are the cells. If we want to change the color, we can simply click on the color dot next to the image label and select a color that we more desire. Within the Analysis Parameter Calibration menu, there are 6 parameters that need to be defined to analyze our fluorescence image, as we can see here. These are going to be similar parameters for all Athena applications analyzing fluorescence images. Of the 6 parameters, the 3 on top perform image processing, while the 3 on the bottom will select the objects according to their size. First, I will describe the smoothing parameter, here described as nucleus smooth. Smoothing is a way to suppress noise in our images and enhance edges to facilitate object detection. the size of the smoothing window should be approximately half the size of the smaller objects of interest. In order to estimate this size, we zoom in on the image using the scroll wheel on the mouse or pinching outward if we're using the touchscreen. To estimate the size, we can then drag the scale bar at the bottom of the image and bring it close to the object of interest. Here we see that this smaller nuclei is perhaps 10 to 15 microns across in its narrower dimension. So we'll want to set the smoothing window to be about half of that, so perhaps 5, which is the current value. We can adjust this in different ways. First, by clicking on the field showing the value, here. In this fashion, we get a pop-up menu where we can type the desired number. Accordingly, we see the masks adjust in our preview image. Additionally, we can adjust the slider immediately beneath the parameter value. In this fashion, we can use the plus and minus buttons in order to see the mask change dynamically as we click. The next parameter we'll define is the background subtraction, which is useful in order to remove fluorescence inhomogeneities in the area surrounding our objects of interest. A good rule of thumb for choosing the background subtraction radius is that it be a bit bigger than the average object of interest. So zooming out, we can see that perhaps this nucleus is a bit more average in size. Again, we can see it's perhaps 15 or 20 microns across. And so making it a little bigger, the 30 microns that we currently have is a reasonable size. Similarly, we can drag the slider to increase the background subtraction or decrease it. As a rule of thumb, increasing either the smoothing or the background subtraction parameter will cause the masks to become larger and expand. On the contrary, making less smoothing or less background subtraction will typically cause the masks to contract. The third parameter we will set is the intensity threshold parameter. This intensity is not the raw intensity that we see visualized in our image. Rather, it is the intensity corresponding to the virtual image held in the computer memory after applying both the smoothing and background subtraction operations. For this reason, we need to set the value of this threshold by trial and error. To do so, we zoom out and can pan by clicking on the mouse to move the image to a desired position. We can move the scale bar out of the way. Now we will use the slider in order to change the threshold and see how the masks adjust accordingly. Decreasing the threshold allows for the selection of dimmer objects. We can see, for instance, here, this object is quite dim, and it would be nice to remove that, so we should increase the threshold. Now, all of the nuclei appear to be well selected. The next parameters we will adjust are the object selections that deal with the area of the nuclei. The first parameter that I'm going to adjust is the patch size. This parameter affects the merging of objects that touch one another. We can see an example where this is desired here in this nucleus. This nucleus has been incorrectly split into two separate objects. Each of the two portions of the nuclei are considered patches. One patch, a second patch. By increasing the permitted size of objects after patch merger, I can cause these two objects to be joined into a single nucleus. Typically, the patch size is approximately the same value as the minimum permitted area. Next, we will select the nuclei based on their area. Zooming out again, All of the nuclei appear to be well identified in this image. If we now move to the next field of view, we can check to see how our parameters are performing. In this particular field of view, we can see that there is a small fluorescent speck that is identified and that is undesirable. If we compare the size of this small object relative to the size of other nuclei that are desirable to keep, then we can get an estimation for the minimum area that is permitted for nuclei to have. This object has an area of approximately 50 square microns. We see this by looking into the attributes area immediately below the image preview area. Increasing the minimum area of the nucleus, we are able to exclude this undesirable small object. We also see other nuclei are well selected, and other small nuclei have an area that is larger than this minimum value. For the maximum value, we see that this nucleus is approximately 330. We can go to the previous field of view. This nucleus about 420, indicating that a maximum area of about 600 should be sufficient for our analysis needs. We can continue to iterate parameter selection by moving between different fields of view and different wells until we're satisfied with the objects that are being selected. Next, we will set the parameters for identifying the cellular cytoplasm, which are shown as the green outline here. To do so, we will select the green thumbnail labeled as cell. Here, we see that there are fewer parameters, and the ones that are present are only associated with image analysis, such as smoothing, background subtraction, and thresholding, and not area selection. This is because in the protein expression application, the nucleus is used as a starting point to identify cells of interest. These particular images are a bit dim, so we will use the pixel intensity histogram in order to increase the contrast. Now we can see that the cell outline is not well approximated to what our eye can detect to be the edge of the cells. As a first approach, we can decrease the threshold. Now we can see the cell outline is more closely aligned with what our eye can pick up. In order to bring the mask closer to the proper edge of the cell, we can change the smoothing and background subtraction. In this case, we want to contract the cell mask, so we will use less smoothing. We can also adjust the background subtraction to be approximately the size of the cells. Zooming in, we see that the cells are perhaps 75, 80 micron in diameter. And we can iterate in order to access a parameter scheme where the cells appear well identified. Now we can double check our object identification by visualizing the overlay channel and making sure that everything looks proper and in order. In this visualization, the red channel is not sufficiently represented. Clicking on the overlay definition, we can increase the amount of red present in our image to better detect what the signal will be. We can move to different well and indeed confirm that the parameters we have set appear to be working well. To move forward, we will click on the Next button. This brings us to a screen where we can optionally define subpopulations. In this tutorial, we will not define subpopulations of cells. This topic will be covered in a separate tutorial video. Instead, we'll move straight on to perform the image analysis by clicking Next one more time. This brings us to the summary screen, where within this window, we are able to see a listing of the parameters used first during image acquisition, and scrolling to the right, we can also see the parameters that we have set for analyzing. Below the window, we can see an important button named Save Protocol button. If we click on it, this dialog box appears where we are able to choose the location for where we want to save our protocol file. Again, this protocol file contains both the imaging parameters for acquisition and the analysis parameters, which are shown in the summary screen to the left. We can save it to the shared folder, which is common to all users, or we can browse to a new location or share it within our user folder. We can select and rename the protocol file, and optionally choose to lock it to prevent future overwriting of the file. We click on Save, and the last thing we need to do is set the name of our dataset. Optionally, it can be defined on the Analysis Setup menu within the Dataset Info button, or we can adjust it here. The name of this dataset, similar to those for Hermes, cannot be changed once they are defined and after analysis is performed. For this tutorial, I will leave the name as it is currently described and simply begin the analysis by clicking Start. Now we can sit back, relax, and watch the images pop up as they're analyzed. It gives us a chance to check the analysis parameters we set to make sure they're identifying the cells properly. Here we arrive to the results screen after the analysis. In the next video, we'll describe how to navigate through different options of the results screen to explore your data.